Welcome to The Explainer. Today we are jumping in a time machine and heading back to 1985. We're going to dig into a really foundational scientific paper that for one of the first times created a detailed map of where serotonin actually gets to work inside the human brain.
You know, before all the fancy imaging we have today, this was a massive mystery. I mean, scientists could study serotonin in animals, sure, but the human brain? That was pretty much a black box. A team of scientists, though, they used what was then some seriously cutting-edge tech to find out, and they zeroed in on two very specific types of serotonin receptors.
Okay, so let's get into it. The main goal here was to find, to characterize, and then to map the exact locations of two major players in the serotonin system, the 5-HT1C and 5-HT2 recognition sites. You can just think of these as specific docking stations for serotonin.
So the researchers really wanted to understand the unique properties of these receptors in us, in humans. And maybe even more importantly, they wanted to see how they stacked up against what they already knew from studying animal brains, like from rats and pigs.
Right, so how in the world do you see something as tiny and, well, invisible as a receptor? You need a pretty clever toolkit. And the scientists on this paper used two really powerful techniques to basically make the invisible visible.
Let's think of it like this. Imagine you've made a special radioactive key that only fits one specific type of molecular lock. Well, you can release these keys into a sample of brain tissue, and then you can literally count exactly how many of those specific locks are there. Pretty smart, right?
Okay, so this next technique takes that counting idea and kicks it up a notch. Autoradiography basically creates a photograph using radioactivity, so you get a literal map that shows you the precise location and even the density of all those receptors.
And boom, this is what that map actually looks like. It's an incredible illustration. What you're seeing here is a slice of the human hippocampus, a part of the brain that's absolutely critical for memory. See the darker areas? The darker it is, the more of those radioactive keys found their locks, which just means there are more receptors packed in there. So now that they have these amazing tools, the first thing the researchers had to do was figure out the basic properties of these receptors, and they decided to start with 5-HT1C.
To figure this out, they ran what are called competition experiments. They basically wanted to see which chemical compounds were the strongest, which ones could push out that radioactive key and take its place, binding most tightly to the 5-HT1C receptor sites.
Now this is really cool because it visualizes that whole competition. Each of these curves is a different drug. And here's the trick. A curve that's further to the left means the drug is more potent. So you can see right away that a drug like mianserin, which is labeled MIAN, is way better at binding than the others. It's even better than serotonin itself, which is labeled 5-HT.
This is a really crucial question in science. Are animal models actually a good stand-in for human biology? So to test this, the scientists directly compared what they found in the human brain to data they had from the pig brain.
Wow, okay, this graph is incredibly telling. Along the bottom, you've got the binding strength in the pig's choroid plexus, and on the side, the strength in the humans. And just look at that. The data points form an almost perfectly straight line. That shows an incredibly strong correlation, and it's a huge clue that the 5-HT1C receptor is remarkably similar between pigs and humans.
So, having gotten a good handle on 5-HT1C, the researchers ran the exact same kind of cross-species comparison for the other receptor, 5-HT2. This time, they checked the human brain against both rat and pig brains to see how it all lined up.
And yep, once again, the visual evidence is it's really compelling. I mean, look at these plots. The data points, again, cluster super tightly along a line, especially if you look at the human and pig brains on the bottom. This is great confirmation that, just like the 5-HT1C receptor, the 5-HT2 receptors are also highly conserved across these species. They just haven't changed much over evolutionary time.
And if the graphs aren't enough, this table really spells it out with hard numbers. It quantifies that similarity. Let's just look at the top row for a drug called cotanserin. The binding values are so close across all three species. It's just hard data that backs up everything we just saw in those graphs.
All right, and now for the big reveal. This is my favorite part. Let's get to the most stunning part of this research and see how it all comes together. The actual photographic map showing where these receptors live inside the human brain.
Whoa, okay, the results for 5-HT1C were just unmistakable. These images are from the choroid plexus, which helps make our cerebrospinal fluid. And those dark areas? That shows an absolutely massive concentration of 5-HT1C receptors. It's practically glowing with them.
The area is just saturated.
Now compare that to this image. This is the 5H2 receptor in the hippocampus, that critical memory area we talked about. You can see right away the 5H2 receptors are not nearly as dense, but they're clearly there. And what's more, they're organized in these really specific layers like the CA1 field. That's a key part of the hippocampus's whole memory circuit.
And if we move to the frontal cortex, the center for all our complex thought, we see a pretty similar pattern. The 5-HT2 receptors show up in these distinct, organized layers, especially in what's called layer 3, which you can see pointed out by the arrow. This isn't random at all. It's a highly structured, very deliberate distribution. And hey, to really put that difference in density into perspective, the concentration of those 5-HT1C receptors in the choroid plexus was measured at over 1,700 units. Now, for comparison, the density of 5-HT2 receptors in that same area, it was less than 20. I mean, that is almost 100 times more concentrated. Just a massive, massive difference.
So, okay, after all this incredibly detailed work, after developing these beautiful and intricate maps of the brain's inner workings, what did it all actually tell us? What's the bigger picture?
Well, here's the bottom line. This study provided for the very first time solid, concrete evidence and a visual guide to where serotonin actually does its job through these two super important pathways. It wasn't just a theory anymore. You could literally see it.
And why this matters so much is that it proved you could directly and precisely map these delicate chemical systems in the one organ we want to understand the most, the human brain itself. This was a huge methodological breakthrough. It just blew the doors open for understanding how these systems can go wrong in disease and, maybe, how new drugs could one day fix them.
You have to remember, this was 1985. This foundational work, by providing a literal blueprint of serotonin's pathways, really did pave the way for decades of research into serotonin's role in everything from mood and memory to the development of treatments for all sorts of complex neurological disorders. It truly helped put us on the map.